There are two types of lambda vectors, replacement vector whereas upper fragment can be replaced by them, or an insertion vector which is a single restriction site. The phage DNA is cleaved with the restriction enzyme that cut it only once and the target is inserted to the site. No phage DNA is removed, therefore much lower size of DNA can be inserted in the case of insertion vectors. Next is the advantage over plasmids. Phages, how they are more advantageous than plasmids. Phage vectors are more efficient than plasmids for cloning larger DNA fragments. Largest clone insert size in a lambda vector is 20 to 23 kb, while that of a plasmid is only 10 kb. Phages are more easy to screen. A large number of them can be screened than the bacterial colonies. Plasmid vectors have to be introduced into bacterial cell by special methods, while phage vectors are directly tested on the bacterial lawn. Bacterial lawn means agar plate bacterial growth, continuous growth കാണിക്കുന്ന ഒരു ഇതാണ് bacterial lawn എന്നാണ് അതിനെ പറയാം, where each phage particle forms a plaque. Plaque is a clear bacteria free zone. അപ്പോൾ അതുവഴി നമുക്ക് ട്രാൻസ്ഫോർമേഷൻ പെട്ടെന്ന് തിരിച്ചറിയാൻ കഴിയും. പ്ലാക്ക് ഹൈബ്രിഡൈസേഷൻ നെക്സ്റ്റ് കമ്മിങ് സ്ലൈഡ്സിൽ എക്സ്പ്ലെയിൻ ചെയ്യുന്നതാണ്. ഇനി അടുത്തതായിട്ട് കോസ്മിഡ്സ് എന്ന് പറയുന്നത് ഹൈബ്രിഡ് വെക്റ്റേഴ്സ് ആണ്. ദേ ആർ ഡിറൈവ്ഡ് ഫ്രം പ്ലാസ്മിഡ്സ് ഹാവിങ് എ ഫ്രാഗ്മെൻറ്റ് ഓഫ് phage lambda DNA. Cosmids are essentially plasmids that contain 250 base pairs of lambda DNA which include the cos site. Cos site എന്ന് വെച്ചാൽ cohesive end ഉള്ള ഭാഗങ്ങളാണ് cos site എന്ന് പറയുന്നത്. The sequence is needed for binding and cleavage by terminase so that under appropriate condition they are packed in vitro into empty lambda phage particles. A typical cosmid has replication origin, unique restriction sites, selectable markers from plasmid. They have a length of 5 kb. The upper size limit for foreign DNA inserted into cosmid and packaged into phage head is very large, 45 kb. Okay, large inserts നമുക്ക് introduce ചെയ്യാം. The protein coat of lambda phage can accommodate any fragment of DNA. The size ranges from 38 to 52 kb. This fragment lies between the two cos sites. Once inside the host cell, the 12 base pair cos ends which were cleaved during the in vitro package base pair and enable the linear DNA to circularize. The circular form of DNA is stable so that the clone DNA can be maintained as a plasmid insert DNA construct, because vectors contain a complete set of plasmid functions. The plasmid part enables them to replicate as it has origin of replication. Plasmid part also helps in selection due to the presence of marker gene due for drug resistance. Their lambda part, that is cos sequences, allows them to be packaged in a phage coat and to be transduced to a recipient by the lambda infection machinery. It has no genes for viral protein, therefore the viral particles are not formed in the host and no cell lysis occurs. ഇപ്പോൾ കോസ്മിഡ്സ് എന്ന് പറയുന്നത് ഒരു ഹൈബ്രിഡ് വെക്റ്ററിൽ പെടുന്ന ഒരിതാണ്. അതിൽ അബൌട്ട് 250 ബേസ് പെയർ ഓഫ് ലാംഡ DNA കൂടിയും പ്ലാസ്മിഡിനോട് കമ്പൈൻ ചെയ്തിട്ടുണ്ടാക്കുന്ന ഒരു വെക്ടറാണിത്. ഇതിൽ മെയിനായിട്ട് കോസ്മിഡ് എന്ന പേര് വരാൻ കാണുന്ന തന്നെ ഇതിൽ കോസ് സൈറ്റ് ഉണ്ട്. There are also sequences needed for binding and cleavage by terminase enzyme. പിന്നെ origin of replication ഒരു വെക്ടറിനെ പോലും ഇതിലുണ്ട്. അതിന് റെസ്ട്രിക്ഷൻ സൈറ്റ്സ് ഉണ്ട്, സെലക്റ്റബിൾ മാർക്കേഴ്സ് ഉണ്ട്, ഇതൊക്കെ ഇതിൻ്റെ ഭാഗങ്ങളാണ്. ഇതിന് അഡീഷൻ ടു ഓൾ ദീസ് ദർ ഇസ് ദ കോസ് സൈറ്റ് പ്രസൻറ്റ് ഹിയർ ആൻഡ് ഇറ്റ് ക്യാൻ ക്യാരി അബൌട്ട് 52 കിലോ ബേസ് പെയർസ് ഓഫ് DNA ഇൻസേർട്സ്. അതൊക്കെയാണ് ഇതിൻ്റെ പ്രത്യേകത എന്ന് പറയുന്നത്. അടുത്തതായിട്ട് ഫേജ്മിഡ് എന്ന് പറയുന്നത്, ഫേജ്മിഡും ഒരു ഹൈബ്രിഡ് വെക്ടറിൽ പെടുന്നതാണ്. ദേ ആർ ഓൾസോ കോൾഡ് പ്ലാസ്മിഡ്സ്. ദേ ആർ derived from plasmids that contain origin of replication for single stranded DNA containing bacteriophage such as F1 or M13. ഇതാണ് ഫേജ്മിഡിൻ്റെ പ്രത്യേകത. പിന്നെ വരുന്നതാണ് വൈറസുകൾ. Number of plant virus like cauliflower mosaic virus, the tobacco mosaic virus, Gemini virus, single stranded DNA വൈറസസിൻ്റെ എക്സാമ്പിൾസ് ഒക്കെ ആയിട്ട് വരുന്നതാണ്. അപ്പോൾ അതും ഒരു നമ്മൾ വെക്ടേഴ്സായിട്ട് യൂസ് ചെയ്യാറുണ്ട്. Animal virus like simian virus, bovine papilloma virus are also used as vectors. TI plasmid of Agrobacterium tumefaciens is used as vectors for plant cells. The advantage of using virus vector is that virus vectors can introduce gene directly by infecting the host cell. It serves as vectors that is used as vectors are powerful promoters which are very much needed for gene expression. They have a capacity to replicate the genome into large numbers. This will help to have high level of gene expression. Certain animal viruses naturally integrate their DNA into host chromosome. This helps the propagation of viral genome throughout the cell line. Next is YAC vector, that is yeast chromosomal artificial chromosome. Yeast artificial chromosome. It's a linear DNA segment which contain all molecular components required for replication in yeast, where DNA segment for several thousand base pairs, about 1 MB, can be cloned. The YACs are linear plasmid vectors that behave like yeast chromosome. Each YAC occurs in two forms. The circular ones grow in the bacteria, linear one in the yeast. A typical YAC consists of centromere elements for chromosome segregation during cell division, telomere and origin of replication from bacteria and ARS of the yeast origin, SUP4 a selectable marker where DNA insert is integrated, although there have been some problems for insert instability. YAC is the most sophisticated yeast vector and to date represents the largest capacity vectors available. They have centromeric, telomeric region, and recombinant DNA is therefore maintained essentially as yeast chromosome. For cloning purpose, YAC is digested with restriction enzymes BAMH1 and EcoR1. The left arm and right arm become linear with the end sequences forming the telomeres. The foreign DNA is cleaved with EcoR1 and YAC arms, and YAC means YAC arms, and the foreign DNA are ligated and then transformed into yeast host cell. The cells that contain YACs can be identified by red or white color selection. Non-transformed yeast contain white colonies. Red colonies of yeast contain recombinant that is YAC molecules. Other artificial chromosome vectors are bacterial artificial chromosome or BAC, for coloning in bacteria, that is mammalian artificial chromosome MAC, human artificial chromosome HAC. MAC and HAC are used for mammalian and human cells. So it consists of centromere ligaments, chromosome segregation, telomere, ori, ARS, and a super selectable marker, that is SUP4. It has they are sophisticated yeast vectors. It is digested with restriction enzymes. Foreign DNA is cleaved by EcoR1 and YAC arms, and the foreign DNA is ligated transformed to the yeast host cell. It is detected by red and white screening. Other examples are BAC, MAC, and HAC. Next, coming to the interesting part of shuttle vectors, the prokaryotic vectors cannot exist and work in eukaryotic cells because the system of the two groups vary. The prokaryotes lack introns while the eukaryotes have intron. Therefore, several vectors have been constructed which may exist both in prokaryote and eukaryote. Such vectors possess two origins of replication. The shuttle vector is a vector constructed so that so that it can propagate in two different host species. That is the importance of shuttle vector. That is, it can exist in both prokaryote and eukaryotic host. So that is the importance. Now we have checked that what is PBR 322, what is PUC 19. Then we looked at what is a cosmid, phagemid, and shuttle vector, and yeast artificial chromosome as vectors. So all these are examples for different kind of vectors, including viruses.